So let's look at biochemistry, and in the biochemistry part, I must tell you a few things before beginning. Biochemistry, for biochemistry there are two different sections actually. One is the mathematical section, another one is the theoretical section. Because they will ask many mathematical questions, especially from the chemistry, hardcore chemistry itself. And few parts only left for the theoretical questions from biochemistry. Now we will solve biochemistry mathematical questions at the end of the course, while now we only go for the theoretical aspects of biochemistry. And in the theoretical aspects, there are few things. I can divide biochemistry in three different parts. One is the structural biochemistry, then the metabolism, and when I mean metabolism, I am talking about the carbohydrates, carbohydrate metabolism. And the third thing, so these are the main things, structural biochemistry and metabolism. These are the two things that are mostly important. In the structural biochemistry, there will be like DNA, RNA structures and their function, Ramachandran plot for the proteins, hydropathy plot, structure of the proteins, and as well as like different types of proteins, globular proteins, fibrous proteins, examples, how they are made up of, what are their structures, and the hemoglobin oxygen saturation curve. These are the things which are really important topic in structural biochemistry. Protein domain, protein motifs, what are the differences, what are the examples of domain and motif, and stuff like that. But mostly they will ask you application based questions, like if you're thinking of a proteolytic enzyme, then or peptidase enzyme which will cleave whether after a specific amino acid or not. So they will give you a sequence of peptide chain, and they will tell you like trypsin, if you treat it with trypsin, how many fragments you will generate, questions like that. Okay, those are the things which are mostly mathematical. Those are also like application based questions that they'll ask. So for biochemistry portion, I will never encourage you to read in details or in depth for the Leninger or White and Void in details, because it will take and consume a lot of your time, and you will get few questions from that only. Because biochemistry, they will ask many questions, but most of the questions are hardcore physical chemistry based questions. That means the del G calculations of the reactions, as well as radioactive decay type of questions, pH calculation questions, buffer problems, questions like that. And amino acid titration based problems, those are the problems that they focus more in biochemistry. Like if they ask you, like in group C, if they ask you 6 questions of biochemistry, 4 will be like that, 2 will be hardcore theory based questions. And even among these 2, they will ask question linked with different biological techniques. Like they'll ask you a question about a polypeptide chain, you treat it with trypsin and load it into the gel. What kind of band pattern in the SDS page, what kind of pattern of the band you can see. Questions like that are more being asked from this region. Okay, so keep that thing in your mind. Like it's not like biochemistry, if you want to read thoroughly from any biochemistry book, any standard biochemistry book, it will consume a huge amount of your time, but you will get 2 to 3 questions to answer from there. So that is up to you whether you will invest your time in biochemistry reading in that sense or not. But it's better that you keep practicing some of the questions, which we will do obviously further in the biology math problem sections. But at this point of biochemistry preparation, I'll get you to know about all those regions which you need to know. So if you at least know the topics that we discussed today for theory, it will be enough. And if you're reading metabolism, try to read it from Leninger. It's a very well written book for the metabolism. For the structural part of the biochemistry, White and Void is better compared to the Leninger. Okay, and for metabolism part, it's better to read biochemistry and metabolism of carbohydrates mostly. You don't need to go for the lipids or amino acid metabolism much often, just the urea cycle if you get some time. Otherwise, carbohydrate metabolism is a primary source of questions. Let's start with it. So today I will discuss a few things about like enzymes and structural biochemistry regions. And again, in the enzymes, they can ask you questions from the enzymes which will be mathematical questions. They will ask you the calculation of turnover number, questions like that in the mathematical part of the preparation. While in the theoretical part for the enzymes, the thing that you need to know is simply the inhibition of the enzyme, different types of enzyme inhibition, what are their properties and what are their differences. That is what you need to know. But before going in there, I must say a little bit about enzymes and how enzymes function. I hope most of you know how enzymes function, right? Like, what are the benefits of utilizing an enzyme for a chemical reaction? If I ask you this question, I think most of you know. Now, the thing about enzyme is that enzyme helps to lower the activation energy, right? Because if you look at a substrate, this is the enzyme and this is the substrate. The idea is, if you give the substrate and allow the substrate to be converted into the product, the activation energy required is huge. That means, you know, this is the reactant. In this specific graph, you will see in the x-axis the course of the reaction, in the y-axis the free energy. And what we know is that in any reaction, we need to have a free energy in the negative value, which will allow the reaction to move forward. Otherwise, that reaction will never move forward. So the free energy change here is going to be negative. But normally what happens, the reactants placed here, the product's free energy ranging very further below. So what they need to do, they need to utilize a specific energy, to utilize that energy to cross a energy barrier to reach the production of the product. And the energy required for that is really high without the utilization of an enzyme. But whenever you utilize an enzyme, we see that energy barrier becomes much smaller. So the amount of energy required to create a total net change of free energy in the minus or the negative scale can be achieved much faster. And that is the beauty of utilizing any catalyst. And enzymes are catalysts that helps to fasten this process and give you the product. And especially why it reduces the energy barrier, it's because when the enzyme is involved in interaction with the substrate and it forms a third different molecule, because an enzyme is a separate molecule, substrate is a separate molecule. When enzyme is attached to the substrate, it forms a third separate molecule known as enzyme substrate complex. And this enzyme substrate complex helps the substrate to be properly placed in the enzyme in a way that not only one substrate, but if the reaction is involved with multiple substrates together even, they'll bring it so close to each other so the reaction can take place really fast. And that is the benefit of utilizing enzymes. So that is the idea. So you have enzyme and substrate, we have a specific free energy, and the product obviously free energy much less. Now without the enzyme, the energy required to reach this product will be much higher. But with the help of enzyme, it is much smaller and shorter. Let me just tell you for very simplest idea now, but during the math class I will explain it details. But the idea says here it's simple. You know that the K-cat is also known as this turnover number. You know, turnover number means the maximum number of substrate molecules converted to product per second by each active site of the enzyme. That's known as the turnover number. And the unit is substrate inverse. Okay. So turnover number means that number of substrate molecules converted to product per second per active site of the enzyme. And catalytic efficiency is known as this K-cat by Km divided by the affinity of the substrate towards the enzyme. That is the catalytic efficiency. Because catalytic efficiency is composed of two components. One is the turnover number, that is how many number of substrates converted to the product per second per active site of the enzyme, divided by also the affinity of the enzyme towards the substrate. So these two components will be checked to form what is known as catalytic efficiency. So the less the Km, the higher the catalytic efficiency. So the less the Km means less substrate is required to reach maximum Vmax.
That means higher affinity. So more the affinity, higher the catalytic efficiency. More the turnover number, higher the catalytic efficiency. Got it? And we can easily calculate kcat by the formula Vmax divided by total enzyme concentration. Maximum velocity divided by the total enzyme concentration will give us the idea of kcat. So we can represent the catalytic efficiency as Vmax ET into Km. Right? That's an idea. So when we'll solve problems, you will see the practical applications of the formula. But that's an idea. Okay? And if we reverse the value of kcat, it will give us the time, the amount of time required for the enzyme to convert one substrate molecule to the product. So in reversal of the turnover number gives us the time required for the enzyme to convert one substrate into the product. So either you know kcat or 1 by kcat. 1 by kcat gives us the time required to convert. Kcat gives us the total number of substrate molecule will be converted to the product per second per each active site. See, let us draw from here. In the x-axis we have substrate concentration, in the y-axis we have the velocity. So what we'll get something like this. So if we continue to add the data and form a plot, it will look something like this. It means, if we continue to rise the substrate concentration, the velocity, the initial velocity, that is V0, also going to rise. So theoretically it will rise till eternity, but practically it should stop, because once the enzyme reaches a certain velocity, beyond this, even if we increase the substrate concentration, the velocity is not increasing. Even if we increase the substrate concentration, the velocity is not increasing. So that velocity till which the enzyme can function is known as Vmax, or maximum velocity. From where we'll get an idea of Vmax. Now if we go somewhere middle where we reach half of the maximum velocity, and if we drag this to the x-axis, we'll also find an intersect at the x-axis. This point in the x-axis is known as Km. Km is the velocity, Km is the substrate concentration at which half of the Vmax is achieved. Okay? And this Km gives us a measure of affinity of an enzyme towards its substrate. It gives us an idea of affinity of an enzyme to its substrate. What is Km? Look at here. In the y-axis we are going half, because half of this Vmax. So this is the total Vmax. So half of this Vmax, and we'll also drag this line to the x-axis. We find this intersect, that is the Km. So Km is what? A substrate concentration for which half of the Vmax can be achieved. Right? Substrate concentration for which half the Vmax is achieved. That is the Km. That's the value of Km. Okay. And it's definitely to deal with the affinity of enzyme towards this substrate. The affinity of enzyme towards this substrate will always change, because remember one thing, if Km is high, what does that mean? If Km is high, what does that mean? Less affinity, because Km high means what? You need more substrate for the enzyme to reach half Vmax. And if Km value is less, that means you need less substrate to reach that same half Vmax. So less Km, high affinity, more Km, less affinity. Got it? either substrate can bind or inhibitor can bind to the active site. Vmax is the same, Km is increasing. In case of non-competitive inhibition, the inhibitor can bind with substrate, the inhibitor can bind with enzyme, or inhibitor can bind with enzyme substrate complex. In uncompetitive, the inhibitor binds with the enzyme substrate complex here. Okay, so what happens in three separate cases, that in competitive inhibition Vmax can be reached at very high substrate concentration and the effect on Km increasing. Effect of Vmax inhibitor, in non-competitive inhibition the effect of Vmax decreases, in case of uncompetitive also decreases, but the value for Km in case of uncompetitive decreases, for non-competitive unaffected. Okay, so for these three separate types of inhibition, the inhibition graph is also different. For competitive you can see Vmax is the same. So Michaelis-Menten is not well to give you a data clearly regarding these inhibitors, which is given by the Lineweaver-Burke plot. For non-competitive, the separate graph where all they are intercepting in the x-axis, not in y. In non-competitive, in uncompetitive, intercepting differently both in x as well as in y axis. Now, two more terms that I should discuss here: isozymes and allozymes. Primary structure, then this primary structure of the protein, they will interact with themselves. The amino acids will interact. They mostly interact with this help of their hydrogen bonding with themselves, especially with the hydroxyl of one side and the oxygen from the other, carbonyl carbon, hydroxyl from one side. So they'll interact with themselves, form hydrogen bonds between themselves to form what is known as a secondary structure. And there are two types of secondary structure: alpha helix and beta sheet. There are different versions of the alpha helix, but there are different versions of the beta sheet, parallel and anti-parallel, but the term is the same. Secondary structure means while the primary structure of the amino acid, they will interact with themselves. Tertiary structure. Tertiary is three-dimensional structure of the proteins. When the secondary structures are formed, then those secondary structures will interact with themselves. Secondary structures will interact with themselves. Okay, like alpha helix or beta sheet, those structures will interact and they will form this tertiary structure. And then the quaternary structure. Tertiary structure is the state which is fully functional. You know, primary amino acid sequence is not at all functional as an enzyme or as a protein. Secondary is not at all. So while secondary structures are interacting with themselves with terms of electrostatic interactions, hydrophobic interactions, mostly hydrophobic interactions, which creates structures of the protein either globular or fibrous. In either of these terms, the tertiary structure is the primary functional unit of the protein. And then there are more hierarchy to this structure. While this multiple tertiary structure of the proteins, they will be linked with one another to form the quaternary structure. And this quaternary structure are three-dimensional. Tertiary is also three-dimensional, but quaternary is when multiple subunits are linked together and they will also function. And the example of quaternary structure is hemoglobin, where you will see two alpha unit and two beta units. So those alpha and beta units separately is functional, but they also interact together to become much effective as a function together. So this is primary structure. The secondary structure of alpha helix and beta sheet, if you look at here, this is alpha helix and this is beta sheet. The interaction pattern is different in terms of how the hydrogen bond is formed. But in all this case, hydrogen bond is the key player. See the hydrogen of NH and the oxygen of CO. This is how the hydrogen bond is formed in all this case. The same thing here as well. But the pattern of hydrogen bond formation is different. The pattern of hydrogen bond formation in alpha helix is different than the beta sheet. So alpha helix, if you look, it looks like a telephone cord structure. See, CO, so oxygen of CO and H from NH interact together. Okay, they form this. So the formation of the bond is always like I and I plus 4. So the first amino acid and the fifth amino acid. The first amino acid, carbonyl carbon here is there, so oxygen of the carbonyl carbon, and the fifth amino acid, hydrogen from the NH group. They will form the hydrogen bond together. So this process continues. That's how it forms alpha helix. In beta sheet, the process is different. In beta sheet, instead of forming the bond in the gap, like 1 with the fifth, that's in the alpha helix, and while they're forming the bond 1 with the fifth, they're also forming the spiral structure. In beta sheet, the idea is different. In beta sheet, carbon, the oxygen from there and NH interact together, side by side, but not between 1 and 5th, between 1 and let's say 30th, 20th, or someplace distantly. That's how the structure is formed. So let's say this is how the structure is formed, the amino acid chain continues. So this one interacting with the other one of the neighboring side. Yes, it is known as intramolecular hydrogen bond. But the bond pattern is not near, the bond pattern is distant, 1 with the 25th, 30th. So by this fashion, they will place the amino acid chain linear, in a linear way in this case. Now while we are looking at this structure, while we are looking at the backbone of amino acids, the backbone of the proteins with the different amino acids there, we always have this backbone, that is NC alpha C, NC alpha C. It continues like that, NC alpha C, right, NC alpha C. So always it continues like this, and whenever we are looking at this backbone where we have this formation of the peptide bond, that is the CO and NH, we have this NC alpha C repeated units in the backbone. And this NC alpha C backbone we have, let's assume this is any three-dimensional model. Let me take the arrow. You can see here NC alpha C. So between N and C alpha there is this interaction, the bond. So they can have a rotation around this bond. This is known as the phi bond. Similarly, between C alpha and C, a rotation around this bond is also possible, known as the psi bond. Phi bond and psi bond. So rotation around these bonds are possible. And there is another bond called between the C and N again, known as the omega. But mostly the pi and psi bond is really important because there are different, in N C alpha is the alpha carbon where the R group is attached. So if the R group is bulkier, then the rotation around the phi bond will be altered. If the R group is smaller, the rotation around the phi bond will be much more allowed. The same thing here with this CO or carbonyl group, because here if this amino acid, because this is the backbone of polypeptide chain, and we know the CO will bind with hydrogen of NH of the neighboring amino acid. So again, rotation around this bond psi also is being regulated. It depends on how the structure of the polypeptide will be formed. So knowing phi and psi angle is really important. If we know the phi and psi angle, we properly know how the polypeptide chain is going to form the structure. Okay, we can predict the structure of the protein by looking at the phi and psi angle. An experiment being conducted with multiple or varying the phi and psi angle to check whether a particular phi psi combination favoring the formation of alpha helix or favoring the formation of beta sheet. If it favors the formation of beta sheet, then whether it favors the formation of parallel or anti-parallel sheet. If it favors the alpha helix, then whether it favors the left-handed helix or right-handed helix, and how exactly the formation is done. So the plot have created by extensive research by G. N. Ramachandran, C. Ramakrishnan and V. Sashishekaran to visualize the energetically allowed region for the phi and psi angle to create the backbone of the polypeptide chain. Based on the values of those phi and psi angles, the structure of the polypeptide either will become alpha helix or beta sheet. it will become parallel or anti-parallel sheet. It will become right-handed or left-handed alpha helix. So this will be very much beneficial if you are working with an unknown amino acid sequence. Let's say we just discovered the sequence of amino acid for a polypeptide chain, and now we want to figure out what kind of protein structure it's going to make. We can easily predict that by finding the phi and psi value of that particular backbone of that amino acid. Because if we look at the Ramachandran plot, it's a quadrant analysis plot. Sorry, let me erase this one. So it's just like a quadrant formation from the middle, like this. So 0 in the center in both this side. Okay, so this is towards positive, negative, same thing, positive, 180, negative 180, because the rotation is possible maximum 180 degree around the phi and psi angle, right? So what we are looking from 0, so you can see here, let's take, so in the y axis we have psi, in the x axis we have phi. So let's take high psi and low phi, positive psi and negative, positive psi and negative phi will place somewhere here. That will form beta sheets. Beta sheets always favor positive psi and negative phi angles. Similarly, negative psi and negative phi favors alpha helix and right-handed alpha helix, while positive phi and positive psi represents left-handed alpha helix. Okay, so this is just an idea of knowing exact phi and psi value. We can predict whether that polypeptide chain is going to form an alpha helix, precisely, preferably whether it's going to form an alpha helix or a beta sheet. I just explained what those 4 quadrant represents. And this idea of the phi and psi angles not only help us to predict the structure, but also it can help us to find out the relative frequency of amino acid that is more preferably be present in either alpha helix or beta sheet, because there are particular amino acids which tend to produce better alpha helix, there are amino acids which tend to produce better beta sheet. For an example, glutamic acids frequency to be present in alpha helix is high compared to beta sheet. Leucine, again similar between alpha helix and beta sheet, almost similar. Threonine present mostly in beta sheet compared to that of the alpha helix. So you don't need to remember that, but remember one thing, that amino acid has their own preferences to be present in either alpha helix or in beta sheet. There are few types of amino acid who tend to present in the same affinity in both alpha helix as well as in beta sheet, but there are few who preferably present more in alpha helix and there are few preferably present more in beta sheet. So that's not the end, because we know the structure, we know the primary structure, and we know how the secondary structure is formed. So once a secondary structure is formed, that's not the end, because secondary structure is not a functional unit. They need to arrange this secondary structure. The secondary structures, like alpha helix, beta sheet, they need to arrange those structures together. And all those secondary structures, so from primary structure it becomes secondary structure, from secondary structure it will become super secondary structure. And the super secondary structure is also known as what is known as the domains of the proteins, and arranging domains will form what is known as motifs of the proteins, and that will be the 3D structure of the protein, and then it will be the quaternary structure of the protein. Okay, so this is the hierarchy of the protein structure. So we know that alpha helix and beta sheets are the unit secondary structure. So two alpha helix together can be a type of super secondary structure. Two beta sheets together can be a super secondary structure. Or beta alpha beta can be a super secondary. Alpha beta alpha can be a super secondary structure. But again, when we have multiple stretch of the super secondary structures linking together, it forms what is known as a motif. So for alpha helix in a stretch, it's the alpha helix motif. It's the alpha helix domain. Then we have beta alpha beta repeated sequences, that's also domain. Beta 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 stretch of the sequence, a domain. But the question is, what is the difference between a domain and a motif? We call that beta alpha beta. This is the domain, but when it will become a motif? When they will arrange properly. For an example, 4 beta will form a Greek key motif. So beta arranging together continuously, it will form what is known as a Greek key motif, which is a key motif structure in immunoglobulin proteins, But let's look at here. Motifs are functional units, but also motif means how the structure is going to arrange. Let's say this 4 beta can be arranged something like this, right? Or 4 beta can also arrange like this. Four beta can also arrange like this. There are multiple ways those 4 beta can be arranged. But when those 4 beta is arranging like this, then it is known as Greek key motif. See here. So domain is 4 beta, beta, continuous stretch of the beta, 4 beta, it's a domain. But it will convert into motif by the way it is arranged. So that's why we have different types of motifs, beta alpha beta motif, this Greek key motif, where we have specific arrangement of this 4 beta structures together. The way they are arranged, that's their motif. Because this structure, the way they arrange this structure, this will be very much beneficial. This structure is required for the function of the protein, for the function of this enzyme. Types of protein structure. There are 2 types of proteins, fibrous and globular. Fibrous proteins, molecules like long and thin molecules. Globular proteins, spherical 3D shaped. Example of fibrous protein, keratin, which is present in here, collagen in skin and bone. Globular protein, hemoglobin, insulin, enzymes. Solubility in water, fibrous proteins are insoluble, globular proteins are soluble. While on the other hand, fibrous proteins don't, they also produce kind of interaction, but the interaction is mostly the other types of interaction. Hydrophobic interaction is also there, but fibrous proteins' role is mostly connectivity in the structures, building the tension strength in the structures, while globular proteins have multiple roles in terms of there are different physical functions played by the globular proteins. For an example, hemoglobin is a very important globular protein that we have. Now the example of the fibrous protein, if you look, the example of the fibrous protein, collagen, the formation of collagen, where we have this DNA producing the mRNA, and from the mRNA translated into the protein products. Once they are producing the protein, there are different stretch of these proteins are produced, different primary structures of the proteins, and then those amino acid sequence, the polypeptide chains, they are linked with each other. They are linked with each other in both the terminal sites and form what is known as pro-collagen. What is known as pro-collagen? Actually, they form what is known as the trimers of the primary sequences. The trimerization of the domain is done. So once trimerization is complete, then what happens? The formation of pro-collagen. And from this pro-collagen they will form collagen, and the process completes. So it's just like, remember the intermediate filaments. We discussed about the structure formation of intermediate filaments, that they don't have any polarity, but they always bind with themselves in the terminal sites, and they'll form thick fibers like that. And those fibers, they will interact with themselves in both these ends. They will interact with themselves in all these sides, and what they form? Collagen fibril. Okay, so collagen is composed of 3 identical left-handed helix, and in the turn they only have 3 amino acids in the turn, and the sequences are glycine, proline, hydroxyproline. So glycine, X is for proline, Y is for hydroxyproline. So they contains approximately 33% of the glycine, 30% of the proline, and 37% of the other peptides. So 63% of all the content of this collagen structure is made up with proline and glycine. So glycine is 33%, and glycine is like 30%, and proline, hydroxyproline is also like that 30%. And the reason they have glycine in it, because proline has a bulkier R group, glycine has the smallest R group. So the only way they can accommodate both without any steric hindrance is including the proline and glycine together. That's how they can easily arrange them together to form this structure of collagen. So in globular proteins, the amino acid side chains are distributed according to their polarities. So the polarities as valine, leucine, isoleucine, methionine, phenylalanine occur mostly in the protein's interior side, because these are all hydrophobic amino acids. While arginine, histidine, lysine, aspartic acid, glutamic acid, they are located in the surface because they become the part of, they interact with the hydrophilic regions. And then serine, threonine, asparagine, glutamine, tyrosine are found in the protein surface, but also in protein's interior, because they are involved in the process of hydrogen bonding, and they always are found during the hydrogen bonding, at a hydrogen bonding state. So again, this idea of hydrophilicity, we can easily get this idea from the hydrophobicity plot or hydrophilicity plot or hydropathy plot. Remember? So remember this very clearly. Remember this very clearly, the positions from highly hydrophobic, isoleucine, valine, leucine, phenylalanine, highly hydrophobic, highly hydrophilic, arginine, lysine, aspartic acid, glutamic acid, which are charged, positive or negative charge, mildly hydrophobic, tyrosine, tryptophan, mildly hydrophilic, histidine, glutamate, serine, those are. You need to remember which amino acids are hydrophobic, which amino acids are hydrophilic, and that distribution is well given again. Basic amino acids means positively charged, lysine, arginine, histidine. Acidic amino acid negatively charged, aspartic acid, glutamic acid. Polar amino acids means hydrophilic, glycine, serine, threonine. Non-polar, hydrophobic, alanine, valine, leucine, isoleucine, all this. So not only you need to remember their properties, you need to know their 3-letter codes as well as their single letter code. It is very, very important to know their 3-letter as well as single letter code, because they will ask you multiple questions from there, and they ask many questions from this protein chemistry, protein structural chemistry. And this protein structural chemistry is very well written in Voigt and Voigt biochemistry. You can also follow the Leninger book because it's well written for everything, but Voigt and Voigt biochemistry is very well written for this protein structural biochemistry. Okay, and we have already talked about the hydrophilicity plot, which is also known as the hydropathy plot, that help us to find out the actual structure of a protein, to predict the structure of a protein, whether the protein will form a globular protein, whether the protein is going to form a membrane embedding protein. And if it's a membrane embedding protein, it's going to form either a barrel shaped protein or a protein interacting at one side of the membrane. We can find it based on looking at their hydrophobic residues in that amino acid stretch in a polypeptide sequence. That's what we are going to find here.